Hello everyone, my name is Xiaoshan Gan and the topic of my presentation is Paracetamol 500mg suppositories. I will discuss about this product from the aspect of biopharmaceutics, formulation and excipient function. In terms of biopharmaceutics, I will first talk about the pharmacokinetics parameter associated with the administration of paracetamol suppository. Upon insertion of suppository into the rectal cavity, the fatty base in the formulation will melt, which results in a release of paracetamol particles from the suppository. The particles will then dissolve in the rectal fluid and get absorbed across the rectal epithelium, which is the first barrier in the drug delivery into the bloodstream. Before distribution throughout the body, paracetamol will undergo significant first-pass effect in the liver, which results in only 68 to 78% of bioavailability with the suppository. The reduced bioavailability is not solely due to the first-pass effect, as oral paracetamol, which encounters the similar metabolism, has a higher bioavailability than suppository. The lower bioavailability may be due to incomplete absorption of paracetamol from the rectal epithelium. After the first-pass metabolism, the active paracetamol enters the bloodstream and distributed into most of the body tissues, including the brain. In the brain, paracetamol crosses the blood-brain barrier, which is the second barrier in the drug delivery to reach CNS. In the CNS, paracetamol binds to and inhibit the COX enzyme, which prevent the synthesis of prostaglandin, result in fever reduction and pain relief. Hence, in the process of drug delivery, there are two main biological barriers that a paracetamol suppository has to encounter, which are the rectal epithelium and the blood-brain barrier BBB. I will first talk about the properties of rectal epithelium and the desired drug properties which favors their absorption across this barrier. Rectal epithelium has a smaller surface area compared to the small intestine, which results in a slower absorption and a slower onset of action for drug administered rectally compared to their oral forms. Hence, drug utilizing the rectal route must not require rapid onset of action. Next, the rectal fluid present in the rectum has a pH of 7.5 and a very low buffer capacity, in which the drug administered rectally will not be chemically changed by the environment. Hence, drug administered rectally need to be in its active form in the formulation. In addition, good water solubility of drug is also required, as the volume of rectal fluid available for dissolution is very small. In addition, there is no active transporter expressed on the rectal epithelium, and this limits the mechanism of substance transport to passive diffusion only. Therefore, drugs need to have a low molecular weight to enable passive diffusion. Lastly, irregular uptake of drug has been observed with suppository, which could lead to variability in the peak plasma drug level and hence the intensity of therapeutic effects. As a result, drug administered rectally need to have a wide therapeutic index, so that the variability in the plasma drug levels will be less likely to cause toxicity with usual doses. Paracetamol has fulfilled all of these desired properties for rectal administration. The suppository form of paracetamol is not to be used for immediate pain relief. In addition, paracetamol is in its active form in the formulation. Moreover, paracetamol has good water solubility, has a low molecular weight, and a wide therapeutic index. The second barrier that paracetamol suppository encounters is the BBB. BBB is an extremely protective barrier due to the tight junction between endothelial cells, minimal pinocytotic activity within endothelial cell, and the presence of active efflux transporter. These features force substances to move transcellularly, either by passive diffusion or active transport to gain entry into the brain. Passive diffusion across the BBB requires the substance to have a low molecular weight and or a high lipophilicity. Paracetamol has a low molecular weight of 151 Dalton. Hence, it is able to penetrate readily across the BBB. In addition, paracetamol is not a substrate for the efflux transporter, which allows its entry into the brain. In terms of formulation, I will first talk about the manufacturing of paracetamol suppository. The first steps involved are the melting of paracetamol into micronized particles to reduce the particle size and the melting of the hard fat. Then, the paracetamol fine powder is mixed with the molten fat until uniform. Then, the mixture is molded into cylindrical shape and cooled for solidification into suppository. Finally, the solid suppository is packaged into blister pack. Now, I will discuss about the clinical performance of suppository compared to tablet. Firstly, the shelf life of suppository is relatively short compared to tablet. Secondly, the bioavailability of suppository is also lower than tablet, which may be the reason why the manufacturer recommends a higher minimum dose, 1000 mg for suppository. However, despite of the slower onset of action for suppository than tablet, similar dosing interval is recommended by the manufacturer. Therefore, tablet will be more effective than suppository in terms of clinical performance, especially when rapid pain relief or fever reduction is required. Other limitations include poor patient acceptability and compliance with suppository and problems with suppository insertion for new users or less trained medical personnel. Due to these limitations, oral form of paracetamol is much preferred over the suppository. However, suppository is useful for those who have difficulty in swallowing the tablets. The formulation of paracetamol suppository is associated with several stability issues, which are mostly caused by the fatty base used in the formulation. Suppositories made up of the fatty base are sometimes brittle enough to suffer damage from abrasion or breakage. Hence, they are usually packed individually in a strip pack to avoid physical abrasion between each unit of drug. In addition, suppositories are particularly sensitive to temperature fluctuation, in which hardening, melting, and resolidification of suppositories can occur due to change in temperature. Therefore, it is very important to store suppository under a constant and appropriate temperature, 30 degrees Celsius. In addition, instead of the strip pack used currently for paracetamol suppositories, I would suggest the manufacturer to use clean blister to enable instant examination of the product appearance for any signs of physical degradation before dispensed by the pharmacist or administered by the patient. In terms of the excipient, paracetamol suppository only contains one excipient, which is the hard fat, and it is very important in facilitating the manufacture and performance of the suppository. It acts as a vehicle as well as a base for incorporation of drugs, which enables solidification during preparation and melting after insertion to enable release of drug. Besides, the use of hard fat instead of the water-miscible base also favors patient requirement, as water-miscible base could cause a painful sensation for the patient due to their hygroscopic effects. In conclusion, in the process of drug delivery from the rectum to the CNS, there are two main biological barriers that paracetamol suppository has to encounter, which are the rectal epithelium and the BBB. Physical-chemical properties of paracetamol fulfill the requirements to overcome these barriers. Addition of excipient is important to facilitate the manufacturing of paracetamol powder into solid suppository and to facilitate the clinical performance and patient requirements upon insertion into rectal cavity. Other formulations of paracetamol are also available in the market, such as tablet, which has a superior clinical performance over suppository in terms of intensity and onset of therapeutic effect. However, suppository is useful for those who cannot make use of the oral route. These are the references that I have used for my presentation and study. And that's the end of my presentation. Thanks for your time.